In this experiment, we looked at the yeast strain Saccharomyces cerevisiae to determine the locus of the lysine 5 or the lys5 gene, which is a major part of the lysine pathway in the genome of this yeast and in the lysine pathway in general. Why is yeast a model? Why did we decide to use yeast as a model for this? Well, yeast is very good because it allows us to visualize and select for the cells that have a certain mutation that would prevent growth. So if certain cells did not have that mutation, they would die, and it would be very easy to see the colonies that did have that mutation. So why are we looking at lys5? Why is lys5 such an important mutation? Well, lys5 encodes for an activator, and this activator causes lys2, or another gene in the lysine pathway, to encode for a reductase. And these two parts come together and they produce a enzyme that catalyzes the reaction of alpha amino adipate becoming alpha amino adipate semialdehyde, and then that goes on to become lysine. What we did was isolate the yeast cells that had a mutation in their lysine pathway, which didn't allow them to produce lysine at all. So why would we do this? Why would we want to isolate those? With those mutant yeast cells, we performed a complementation test. So we really wanted to find those distinct ones in order to look at the genome. We performed a complementation test, which is when we took two known lys mutants. You can see what we did in this diagram here, but I'm just going to show it on the side a little bit larger. So we took two known lys mutants, so lys5 and lys2 mutants that we knew were specifically mutants that did not have lys5 and did not have lys2. The mutants that grew at the intersection with the lys2 mutants that did not have lys2 but did have lys5, that meant that our unknown mutant did not have lys5 but did have lys2. So those were what we were looking for. We were looking for the colonies that grew on the known lys2 mutants. After we identified which mutants were lys5 mutants that did not produce lys5 activator, we sent those particular genomic DNA of those colonies out for sequencing. And when we got them back, we looked at the map of where this lysine mutation was on the yeast genome. So through all this, we were able to pinpoint the genetic basis of the lys5 mutation. And what we found was that there was a substitution. So in a non-mutant, you can see that the original codon was TCA, which produced serine. And serine is a polar uncharged side chain, but in the lys5 mutants, a T in the 125 base pair position was inserted instead of a C, so it was a mismatch. And that mismatch, the TTA, instead of the TCA, produced leucine. And leucine has a hydrophobic side chain, so they really were not exchangeable, and so the functions were not able to happen. So this was unable to happen. This was unable to happen. Even this, because it produced something completely different. So in looking at that, we had an S42L mutation. Now, where would we go from here? What do we use this information to do? So to understand how this mutation affected the lysine production more in depth, for future experiments, we would want to explore the folding patterns of the leucine amino acid versus the serine amino acid. And then that experiment would allow us to see a more in-depth understanding of the folding patterns and how that comes into play. Our references are down here. This is what was used to aid in understanding, and the method was courtesy of Dr. Kaczynski-Cullen and Associates in the Biology Laboratory Manual. Thank you.